Hello, everyone. I'd love to know if anyone's hearing me. Dave Cozano, you hear me? I see you on my screen. Okay, Kathy Alban heard me. Okay, so welcome, everyone. This is going to be truly a hybrid meeting. And I'm Roy Herbst from Yale Cancer Center, currently the PI of the steering protocol and the screening protocol, excuse me, and very happy to be here with all of you. This has been a tradition at our SWOG meetings to have this LungMAP update meeting where we go through and hear about different issues regarding the trial. We hear about existing and in planning sub-studies, and we have a chance to talk and hear from you. I'm very fortunate that working closely with me as the vice chair is Dr. Jyoti Patel from Northwestern, and I'm going to let Jyoti introduce herself.
Thanks so much, Roy, and it's great to have so many people online, and I imagine a pretty full room. Again, it's a little bit tough because we can't see you, but thanks so much for making the time. It's really, I think, an exciting time in LungMAP. We have a lot of studies that are coming down the way, some great translational work, and some updates on studies that have completed accrual. So really a great time to be involved. We look forward to working with all of you and hearing how we can make this even more impactful to our patients.
Great. Now, before we start, I just thought, let's make sure we have the communication. So Janelle Gray, are you there and can we see and hear you?
Janelle is coming up now, Roy.
Mm-hmm.
Okay, and then David, we don't see you for some reason. There's no camera in the room?
No.
So this is something we had earlier as well. Unfortunately, there's no two-way visual, but that's okay. We know your voices. So I had wanted to sort of just thank Karen Kelly. I don't know if Karen Kelly's on the line, our outgoing lung committee chair. Karen's now the CEO of ISLAC, and we miss her already, but we're fortunate to have Janelle Gray now as our lung committee chair at SWOG, and Janelle will say a few words. So David, what do you think, Jyoti? Do you want to moderate from video, or should we have David do it from there, or whatever is your preference, Jyoti?
Sure. I actually think the flow might be better, David, if you don't mind doing it. And then I'll make a plea like I did yesterday. If someone has a phone that they could just kind of prop up, you don't have to walk around, so we can kind of get a sense of how big the room is and who's there. It'd be really helpful too.
We have about 5,000 in the room now, but we're expecting a few more.
So what I would suggest is you do two selfies, one inwardly.
That's right. And we have the press here. So the difference.
And the police, I'm sure, the police too.
The difference, Roy.
The phone face you, so we can see you, David. And then,
so the difference, Roy, is that we don't have any food. So when you were here, it was better. We had a full house when we had food. That being said, why don't we have Janelle get up and make a few introductory comments?
All right. Hi, everybody. Excited to be here with you. Thanks for joining those who are here in person, as well as those who are joining us virtually. Hopefully we'll get to full face-to-face meeting back in Chicago. All right, I think we had a couple of slides. How do we get back to the slideshow? There we go. I think maybe the next slide. We're supposed to point at. Maybe point at.
I'm advancing to point at where? Here?
Maybe. Oh, okay. Thank you. Maybe control now. There you go.
Okay, we're going to introduce the new lung committee chair.
That's me. Hi. So introducing myself. So I'm Janelle Gray. I'm a thoracic medical oncologist. I work at Moffitt Cancer Center. Real pleasure and honor again to be serving in this role. Really big shoes to fill. Want to take a moment, like Roy said, to just to thank and join me in recognizing Dr. Kelly for all of her years of service, and just recognizing her for what she has been able to accomplish for all of our patients battling lung cancer. She has really given us a great foundation and platform from which to continue onward in excellence. And again, look forward to working and partnering with you all. One of the things when I was discussing taking over for Dr. Kelly is, really sitting and listening to Dr. Blanc and what we think we need and where should we be going for SWOG as a whole, in addition to what we should be doing in the lung committee. And number one thing is continue to usher in new treatment paradigms for our patients battling lung cancer. We have done an outstanding job. You're going to hear a lot about these, the trials that are ongoing, as well as the ones that are coming down the pipeline to one of our largest initiatives in the Lung Committee, which is LungMAP and all the enterprise. I still remember when David and Valley presented it at the Lung Disease Working Group, and just to see where it started from that discussion to where it is now. And you'll hear a lot more about that from David today, I hope. It's been really, really impressive and expansive. The other thing I think, you know, we need to always do is take a pause and look at our patient populations. You know, this is a cooperative group, and we have a lot of sites within the national cooperative group system. And in there, we have a very diverse patient population. And so as we were designing these trials and thinking about the trials, and as David says, don't bring the patient to the trial, bring the trials to the patients. We need to keep that in mind, and liberalizing inclusion criteria, thinking about what the needs are across the spectrum of patients that we all took an oath to serve. And I think this is one of the ways that we can do it in the Lung Committee. And also, I'm a big proponent of mentoring. You know, I actually hate being up here. I'm going to be very honest. You can see they made me come up here. I enjoy being in the crowd. I enjoy being fly on the wall. It's really about that next generation, finding those superstars. Who are they? Who's going to be that next person up here on this podium talking to you about what their vision is for the Lung Committee, and making sure that we set those individuals up for success. I think working all together to create a safe environment so that they can present their ideas and feel welcome. So those are just some things that I wanted to share with you. I made this plea at the disease working group meeting earlier today. And if anybody has any junior faculty that they'd like to bring into the fold, I'd highly encourage that. And just please connect me with them so we can find some opportunities for them. And with that, I'll hand it back over.
Okay, thank you,
Thank you.
Janelle. And before I give a brief presentation on the accomplishments of LungMAP, I realize we don't have an agenda to show you, so I'll tell you what's coming up. So we actually have a series of great presentations. I'm going to talk a little bit about what LungMAP has accomplished since its initiation in 2014. Dr. Herbst will come back on to give an update of manuscripts. Dr. Cosino, our head of our Translational Medicine Committee for LungMAP, will talk about what that committee is doing, some of its accomplishments. Dr. Pata will talk about 1900E. I can never remember exactly what all the numbers mean, but you'll hear about these. Dr. Rangel, I know this one, will talk about the latest addition to the LungMAP activated studies, 1800D. Dr. Gray will talk about 1900F. Dr. Haas Borgai will come on virtually and give us an update from the Drug Selection Committee. So as you know, as a public-private partnership, this is quite different from a typical NCTN study. And we have a committee that is, of course, not only SWOG, but all the groups, representatives from the National Cancer Institute, and a variety of other places that help us decide what sub-studies we're going to do. Then we'll hear about the Accrual Enhancement Committee. And lastly, Dr. Jyoti Patel, our vice chair, will come back on and talk about what's in the future. So with that, I'll tell you what's in the past. These are my disclosures. Well, you've seen this slide a lot, and it's still worthwhile showing it at the beginning of any LungMAP meeting, because there is nothing like LungMAP in the world. It was designed to be something unique for the National Cancer Institute and NCTN, and that is a public-private partnership. You can see that it includes, of course, all the adult cooperative groups, but also the foundation of the NIH, who is the purse holder for the finances and really helps us with a lot of the organizational structure. The Friends of Cancer Research with Ellen Siegel, who helped us set this up and continues to be a very active partner within LungMAP. Of course, the National Cancer Institute And the FDA, who of course is not a formal partner because that would be a little bit of a conflict of interest, but highly supportive of LungMAP and works with us very closely. And then you can see here partners. So this is part of the public-private partnership. Foundation Medicine, of course, for our NGS testing platform, initially in tissue, now in both tissue and blood, and a variety of pharmaceutical companies. So in order to know where we are, you have to ask first, why a master protocol in non-small cell lung cancer? There have been many attempts to do things similar to LungMAP, and some of them in other tumor types literally never got off the ground. I will say there is an Alzheimer's master protocol that was designed after they looked at all of our documents about how we set up LungMAP. So the platform, of course, has now been has been adapted for industry by some industry for smaller trials, usually not this large platform like we've got. Then the second thing is, why a master protocol in this disease specifically? And it's because genomically and immunologically, lung cancer, non-small cell lung cancer is ideal for a master protocol. As you can see on the left, it is genomically complex, and the number of oncogenes that we test for goes up on the average about every 4 months. We're adding more, not always a first-line therapy, some are second line, but that is a reason that it's good for a master protocol. And the second on the right, that's from the TCGA, shows that non-small cell lung cancer, both quantitatively and qualitatively, is ideal for a master protocol. It has among the highest of any tumor type. It's on the far right, adenocarcinoma and squamous cell lung cancer. Quantitatively, the most mutations, tumor mutational burden, but also qualitatively, those are these orange C to A transversion mutations, which are highly neoantigenic. So again, a poster child for checkpoint immunotherapy. In 2014, when we designed LungMAP and initiated it, what were our goals? And I show them here. We wanted to test new anti-cancer therapies in uncommon or rare genotypes where everyone, including big industry, were struggling to screen 100 patients for 3 or 4 or 5, which could go on to a trial. But if you screened enough in a master protocol setting with multiple genotypic sub-studies, it was worth doing for the patients and for the oncologists. So check, we've done that. We wanted to develop therapies in the immunotherapy space, and we've completed 2, including a phase 3 study in previously treated, as well as 2 others more recently in IO refractory disease. In 2014, next generation sequencing, this may be hard to believe in 2022, but it was investigational. In fact, in terms of practice outside of a clinical trial, only 2% of advanced non-small cell lung cancer was being genotyped with next generation sequencing. You know, you tested for a few genes, you would do PCR for EGFR, you do FISH for ALK and so forth, but that was about it. So we wanted to transition this to the clinic. We've been highly successful in doing that. And of course, now there are 20 companies that are out there competing doing next generation sequencing. We wanted to do it with an acceptable turnaround time. When this was research, very often the results would come back 28 days or longer after the specimen was submitted. So in the entire history of LungMAP, our turnaround time for getting the information back is an average of about 9 to 10 days. So that is clinically applicable. So check again there. We wanted to be able to expedite the clinical trials process through a public-private partnership. As I've already shown you, we've done that. And as Janelle said, we wanted to flip the paradigm of patients from the community going to a few academic centers to instead bringing these state-of-the-art trials to many sites across the country, community centers everywhere. As I'll show you in a minute, check, we've done that. And lastly, we wanted to take the information we gained from the trials, other than just the response rate or the survival, we wanted to use that genomic and immunologic resource to create new science. And I'll show you some of that that's being done. So this is taking the trials to the patients. I haven't counted all the dots, but I'm told there are over 900 sites throughout the United States which have activated LungMAP. That's the most recent. Over half of our accrual comes from community sites. So this is really a nationwide process. In a survey conducted by the CTEP, they asked sites during COVID, what did you shut down? Were you able to keep your clinical trials going? What sort of prioritization did you have? And over feedback was, we kept LungMAP open. It was a high priority for our patients and for our investigators. So this shows some of these accomplishments. 13 partners, 15 investigational drugs, 16 trials initiated, 14 completed. I won't read all of these, but over 4,000 patients have now been enrolled in the screening portion of the trial. And as you can see, about 1,000 treated with these new therapies. I will also say that this resource is incredibly valuable now for the entire cancer community, 10,000 plus specimens. Shared costs, of course, because of the public-private partnership, industry pays a share, and the National Cancer Institute pays a share as well. For instance, I think some of you know we provide a repeat biopsy. We pay for the biopsy if the patient needs it to get adequate tissue. This is where we are. As you know, initially, this was called S1900. We transitioned to a new name. Mary, how long ago? 2 years now?
January of 2019.
January 2019 to LungMAP. You can see everything that's in dark blue is completed. On the far left, those are the genotypic studies. On the right in blue, the ones in the immunotherapy space. You can see in green what's open, but you can see several in development. And Dr. Borgai will tell you, we've never had so much interest. We have people coming to us almost every week with new proposals. So we're very busy in trying to fill up the spaces here. Dr. Herbst made an interesting comment last year. He said, you know, the store is open, but we don't have much on our shelves right now. And what he meant is we completed some of these studies before we were supposed to. And then they were completed and we didn't have a backup ready to go yet. So now we're going to try to make sure we have plenty of studies because it's the best thing for LungMAP. It's the best thing for our investigators. It's the best thing for our patients. Creating new science. And here I just show 6 examples. I think there were 4 additional abstracts submitted to AACR or ASCO and 1 to World Lung, at least that I know of. So there are new things that aren't even on here, except for the last one I'll mention. But you can see these are some of the things we've done through our translational medicine group led by Dr. Phil Mack and Dr. Fred Hurst, who were seated here. So these are just some examples. I won't actually go through them because we've got a lot to cover today, but there's more coming. And that is, I think, incredibly important that we not only conduct the trials, but we help all of the lung cancer community by learning more about why drugs worked or why they didn't work and how we might do better in the future. And I'll just point out one of these. It's in the bottom middle. That's a study done by Dr. Reese. It's a PARP inhibitor in patients with either BRCA mutated lung cancer or high loss of heterozygosity. And in terms of the BRCA, as you know, PARP inhibitors are approved in several tumor types for patients with BRCA mutations. They don't work very well. Those PARP inhibitors don't work very well in non-small cell lung cancer because it's a heterozygous mutation. It's monoallelic, not biallelic in comparison to the other tumor types. So we're creating science here. And with that, I will close and I will go to Dr. Herbst, who will give you a manuscript update, and I can advance the slides if you'd like, Roy.
Thanks, David. And for those who might notice, I now have a background. I don't know if, can the audience see me or not?
Yeah.
So I have been sent a picture of the room. You have got quite a few people there. It looks like a very spacious room. So hopefully someone's going to turn their cell phone on so that the audience can see. So I'm now going to give a very quick manuscript update. If we go to the first slide, please, Dr. Gandera.
Yeah, I'm trying to advance. Where should I point it? Oh yeah, we can obviously not use this. It's low tech.
Sorry, let's see if it works first.
Isn't Seattle where Microsoft is? They
No.
could- What
We
the heck?
may have to have you advance the slides for us back there, AV.
I guess
Maybe we've been bad and they took away our permission, Janelle says.
okay, so here's so one of the things that's very important is to publish our data, whether it's positive or negative. And thank you to the Ops Center, Statistical Center,
Sorry?
Mary and her team, and all the authors. So I just thought I would show an example, some of the studies that have been published in the last year. Well, this is very bad slides, but we have, you can see the different slides. You can see we just had a reply by Scott Gettinger. The formatting is wrong on these, I'm sorry. And then we have the durvalumab plus tremelimumab in previously treated patients. Natasha, next slide, please, David. Then we had the 1400I, this in JAMA Oncology, the Ipi-Nivo versus Nivo. To this day, it's one of the first studies that compares that doublet versus the single drug. It's in the refractory setting, but IO naive. There's a lot of data mining coming from this, including work with the CMAX, looking at some of the immune biomarkers. There actually are some abstracts at some of the major meetings. Then we've also looked at patient knowledge. This is work by Dr. Roth and Ramsey, Hirschman, and the committee that looks at patient-reported outcomes, patient knowledge and expectations about return of genomic results. And then you can see Tawfiq Coco, phase 2 study of talzoparib in patients with homologous recombination repair deficient squamous cell lung cancer. Next slide. And then you can see the previously treated stage 4 recurrent LungMAP study of PD-L1 inhibitor. This is durvalumab. This was Hasborgai and team published this. Next, you can see the study of telsotunib vedotin. This is, of course, an ADC, patients with c-Met positive stage 4 recurrent squamous lung cancer. Some of these studies didn't meet their endpoint, but they are important nevertheless. And then you can see a very important paper, lead author Mary Redman. And this is a paper you should reference when you're writing about the Lung Master Protocol, biomarker-driven therapies for previously treated squamous cell non-small cell lung cancer. So it's really the whole S1400 experience, talking about the design of the protocol and the biomarkers. Now, on the next slide, this is the coming attractions. So Karen Redcamp is in the room, and 1800A was submitted to ASCO. Now, they'll probably cut me off if they could figure out how to do this if I were to tell you the results, but I'm not going to. But I will tell you that it's been selected for the podium at ASCO, and there's a manuscript in preparation. So keep an eye on that. We have to just keep that embargoed for ASCO, but we'll look forward to seeing those data. And we are going to have an event at ASCO. You'll be hearing Stacey Adams on the phone. You'll be hearing from the FNH, where we'll all get together. And there are numerous other correlative science papers and abstracts. You'll hear from Dave Cozino in a bit, and I'm sure he'll talk about some of that, so I didn't want to steal his thunder. That's really all I have to say, except that We will be pushing you. There are a couple of you out there. I'm not going to mention you by name. You've gotten some emails from me. You've got to write your paper. If you don't, we'll pass it to someone else. We're going to be very strict, coming from the very top echelons of SWOG on getting these data out. We're going to have so much data in the next year, I predict. So that's it. Josie, anything you want to add on that?
No, I think things are moving really well. There's a lot of exciting opportunities to look at subsets in some of these trials. So, please come to us with your ideas about how we can parse the data out. And David's going to take it from here, in which, again, we're really looking for proposals to use this vast repository of clinical and genetic information.
So next,
oh, please.
Oh, actually, I was just going to introduce you, David. So David Cousino, who also created some of that new science I was talking about, is the chair of a translational medicine committee. And so David, please give us an update.
Really so excited to be here to give this update regarding the Translational Medicine Committee. Next slide, please. Here are just a couple of disclosures. Next slide, please. And so the purpose of the TM committee broadly is to bring together expertise in translational medicine to assist in biomarker selection and definitions and sample collection decisions for substudies that are under development, to review proposals for prospective and retrospective translational research, and to assist in the analysis and publication of sequencing and other correlative science data. I should really add that the real purpose then of our work is that we learn from every trial that we do. So the idea is that regardless of what a study may show, whether it meets the primary endpoint or not, we have the opportunity to make the most of each study, given all the effort, the patients who go into these studies, and really wanting to refine what we know about lung cancer so that we can better design studies in the future that could incorporate biomarkers and target treatments to subsets of patients who are most likely to benefit from those novel treatment concepts. Next slide, please. And it's been such a wonderful thing working with this fine group of folks here, representing LungMAP leadership, representatives across the NCTN cooperative groups, the statisticians that are the lifeblood of our analytical teams, the FNIH, given the public-private nature of LungMAP as a whole, but really the glue that binds us together. Members from Foundation Medicine, given the key central component of their Foundation 1 testing for our DNA sequencing. And then Nationwide Biobank, given that it is a collection of these biospecimens from all of these sub-studies and the screening protocols that really are the central resource for ongoing and future analyses in TM medicine. Next slide, please. And so just broadly speaking, a few areas of expertise, thinking through our committee and their academic and other interests. We do have wonderful representatives with expertise in immuno-oncology, in targeted therapies, in DNA repair, and anti-angiogenic therapy. But I should also add that if there are ideas that may be beyond these areas, we can certainly also bring together expertise in any relevant biology of interest. We have those who have thought long and hard about optimal biospecimen banking and processing and pathology, as well as clinical genomics. Next slide, please. And so just as a taste of the translational medicine resources that we have, one very nice example is the sequencing data from that older phase of LungMAP, the S1900 profile portfolio, which spanned the years 2014 to 2019, where there were 1864 patients with stage 4 or recurrent squamous cell lung cancer enrolled, and for whom there is next generation sequencing data from Foundation 1 sequencing available for 1672 patients. This was using a platform that sequenced the exons and or introns of 313 cancer-related genes. And given the number of samples, this is a total of 36,000 variants that have been identified. And thinking through what this means, it's really the largest NGS data set of advanced squamous cell lung cancer previously treated patients that's out there. And so this slide also provides some sense of the number of variants that we found per subject and the types of variants, as you can see here. Next slide, please. And so really, we want people to be able to use data like these. And I tried to simplify this as much as possible, really to get a sense of what a TM proposal might be, and just to quickly get to the sense of whether it's feasible based on the resources that we have. You do not want people to necessarily have to spend a whole lot of time putting together a proposal to learn that something might not be feasible because we don't have the correct biospecimens for the work, for example. And so to simplify this, we now have this really just one page. It can be 2 pages if there's extra info you want to supply, but a letter of intent, which is really just to gather preliminary information on the proposed retrospective use of any bank specimens and/or data. It could be one or both. And here is an email address that will ensure that this LOI, when completed, will get to me and a few other folks who can help triage any translational medicine proposal you have. As you can see from the form, it's really meant to be something quite simple, really revolving around which studies you're interested in, the types of data that are requested, as well as the specimen types and amounts, roughly, that would be needed for the proposal, something about the proposed assays, and any other considerations that you feel upfront would be helpful in evaluating a concept. And this is really meant to be triage with a relatively low bar for invitation to write a full request, but at least this would be a way not to get too far into writing a proposal before discovering, for example, we may not have the samples necessary for your idea. Next slide, please. But since we do have quite a number of different folks that come together, with the whole idea of it taking a village to conduct translational medicine, this is reflected in the review process. And I did want to go over a little bit of this so that it is as straightforward as we can make it. Recognizing that there are different paths for any given idea, this is a general framework where we can start with that LOI to review a concept for basic feasibility. And then if it looks promising, this can be presented to the Translational Medicine Committee, which has expertise to review these proposals. At this point, we could, as one possibility, if this meets consensus approval, have the completion of a full SWOG TM proposal templated form. Next slide, please. But what I really wanted to highlight that it isn't just our committee that ultimately must approve a proposal. We do need our statisticians to weigh in based on the statistical considerations to make sure, for example, that a study would be adequately powered to answer the questions that are being posed. And we also need review by our operations team at SWOG to look at those considerations to make sure, again, that a project is geared for success. Once all 3 parties in, the TM committee for scientific review, the statistical review, and operations review, if all these basically can approve this concept, then they can go on to, next slide, please. Approval by an overall committee, including those from the LungMAP leadership, and the sub-study chairs, if there are specific sub-studies that are involved, who would also be a part of the approval process for a translational medicine proposal. And this is when a final vote within the LungMAP structure would occur. Next slide, please. But it's not quite done here. We do have one more review by our operations group to prepare a protocol amendment as needed. And generally, these would be needed for these analyses, which would then ultimately be reviewed by CTEP to garner their approval, following the standard process for these proposals. And then we would also, at this point, also consider any contracts and funding considerations that we may need to also ensure are proper for the study. Next slide, please. And so this is how it all looks when you put it together. The intent isn't to try to complicate, but rather to try to streamline this process while being respectful of all the different role groups that can ensure that a translational medicine proposal will be successful. Next slide, please. And so, thinking through this past year's proposals, I'm really happy to hear there's been quite active interest in thinking through possible use of our biospecimens. There have been a couple of themes that have emerged and what's felt by the committee and others to be more likely to be a successful proposal. And so here are just a couple tips along those lines. One, the priority for the use of biospecimens, especially when these are of limited supplies, such as tissue and blood-based biospecimens. The CTEP and other organizations generally do prefer validation studies and hypothesis-based research as opposed to purely exploratory analyses. So that's one consideration. Our committee has reviewed a few emerging technologies that have seemed very promising and quite exciting, but it was felt perhaps premature for the use of our precious LungMAP resources, although the hope is that with maturity, these will be very good uses for such samples. On the other hand, there can be a lower bar for certain kinds of analyses, such as secondary use of data, whether that's the genomic data that we've accumulated or digital pathology data, basically inexhaustible data. And so because we can provide these data more widely, their use can also similarly be given wider scope. Now, of course, proposals with funding that's secured or available would be favored. I just want to call attention to one possible source of funding for these kinds of studies. If we go to the next slide, this is NCIFOA here for assay validation of high quality markers for clinical studies in cancer. So basically the UH2, UH3 mechanism. And the UH2 phase comes with up to $275,000 over 2 years, and the UH3 phase $250,000 per year for up to 3 years. And this is an application you can do 3 times per year, same as several other NIH grants. Next slide, please. I just thought this was helpful because it also gives you the kind of sense of what is a very attractive proposal. We'll be including assays whose marker classifier is likely to be used in treatment trials, technologies that are already in use or soon to be approved for use in clinical labs, applications that improve standardization or harmonization of assays among labs for use in clinical trials. And this is all predicated on having a working assay that can use, of course, human specimens. Next slide, please. And just wanted to maybe call out, Dr. Gandara had already done so, Jonathan Reese's wonderful analysis in S1900A. So I won't belabor what he found in this work, the importance of having biallelic changes in the BRCA1/2 genes. But to give you an example of the kind of amendment that our committee can help with, we had recently submitted one to CTEP to further these analyses to really look at reversion mutations and how they may have had a role in these responses, and concordance between plasma and tissue-based identification of mutations in BRCA genes and other homologous recombination deficiency genes. And so as you think through possible ways to interpret the data and are considering additional potential biomarkers that might ultimately speak to response, our committee would be very happy to think through how we can best craft hypotheses for your research. Next slide, please. And so other analyses, Dr. Gandara didn't mention one of his own projects, but this is a really nice one that he's worked on that I have fingers crossed about. But taking a look, at biomarkers beyond TMB and PD-L1, as much as they've served us in looking at immunotherapy responses, adding other, alterations and other phenomena to create a signature that might better identify those patients who are to stand to benefit from immunotherapy. And Suki Pada is doing very fine work right now, looking at the genomic landscape of KRAS mutation subtypes in lung cancer using LungMAP data. So I think that's what I have. Next slide, please. And so just wanted to say thank you.
Thank you, David. And I should add that we will have a question and answer session at the end. I think we've arranged for enough time for that. So we realize we're not taking your questions for individual speakers, but we'll take them en masse at the end. Next, we're going to go to Dr. Suki Pata, regarding S1900E, which you've already heard alluded to. Suki, are you on?
Yeah, thank you, David. I'm here.
Great. Do you want me to advance for you?
Oh, yes, please. Thank you. Appreciate that. Yeah, so I'll give everyone an update on LungMAP S1900E, which is chaired by myself and David Gerber at UT Southwestern. Next slide. All right, so this is just a clinical slide. I think most of us were familiar with the data of sotorasib and previously treated KRAS G12C non-small cell lung cancer, which resulted in the FDA approval in May of 2021. From that exploratory analysis in CodeBreak 100, there were differences in efficacy observed in co-mutation subtypes, looking at specific co-mutations in tumor suppressor genes of P53, STK11, and KEAP1. And I think one of the other messages that was important to take from this analysis that it's also important to account for co-occurring co-mutations, because that's where a signal was observed in STK11 co-mutations, where KEAP1 was wild type, where there may actually be an enhanced response rate and potentially even progression-free survival. Next slide, please. And so I think everyone's familiar with the schema at this point, but basically S1900E is examining sotorasib in robust upfront biomarker-defined KRAS G12C co-mutation subsets, including P53, STK11, and then in other buckets. So patients are treated with standard dosing of sotorasib at 960 milligrams daily. And then also importantly, at progression, we're going to be collecting circulating tumor DNA to identify mechanisms of resistance across all of these different cohorts, but also to examine whether there's any differences within different co-mutation subtypes. The primary endpoint for each cohort is objective response rate. Next slide. So why does S1900E matter in the setting of the fact that we now have sotorasib approved? Well, first of all, it doubles the Amgen CodeBreak 100 sample size, which was at 126. We'll add 105 patients to the literature, in addition to the randomized phase 3 studies that are going on with sotorasib in this setting. As I mentioned before, the biggest advantage of this study is the rigorous upfront co-mutation biomarker definitions, which also accounts for co-occurring co-mutations among P53, STK11, and KEAP1. For example, in STK11 in the LungMAP data set, 80% co-occur with either KEAP1 or P53 or any permutation of that. So it's important to account for those. Based on the prospective statistical design, the objective response rates are expected to have more narrow 95% confidence intervals compared to that was reported in CodeBreak 100. And given the LungMAP screening study, we're hoping this will include a more diverse patient population, which was not observed in CodeBreak 100. Next slide, please. So this is a accrual. We're doing actually quite well. We opened in April of 2021, so it's been about a year. And so 24 out of 40 patients have enrolled in P53, 13 out of 25 in the STK11 cohort, and then 21 out of 40 in the all other cohort. And we are either meeting or are ahead of our accrual timelines. We've had 6 patients enrolled in the last 30 days. I think hopefully to support this ongoing good accrual is an amendment has just been passed, CIRB and NCIFOA approved, which really liberalizes the eligibility, especially in light of the approval. That was very important for us to do. So I think one of the big things as well is it does allow now patients with untreated asymptomatic brain metastases, per the NCI guidance, shorter washouts for prior anti-cancer therapies, other drug-drug interactions. And then we've also, very importantly, removed the interim analysis so that we can just smoothly continue to accrue the study. So thank you for your time and support of S1900E.
Thank you, Dr. Pata. And I'll just add here, I think this study is also important because it shows the additional role of LungMAP. Most people think, oh, LungMAP means you're going to be looking at a new drug or you're going to be looking at a new combination. But even for a drug that's already FDA approved, there's a lot we don't know. And because, as Roy Herbst says, we have a big funnel, meaning we're screening lots of patients, we can accrue very rapidly to studies that will complement what industry is doing and allow us to understand better the biology of who responds and who doesn't and why. Okay, so then we're going to move to John Rengel, who's already given this presentation 3 times, I think, at SWOG, but not for this group. So John. Dr. Rengel, are you on? Well, maybe he thought he had completed his round having given it so much. Here appears not to be. So I can show his slides. This is the study. How many of you know about this study? You're supposed to raise your hand. All right, so everybody should know about this. This was activated about, what, a month ago, Mary?
Excuse me, February 15th?
Okay, February 15th, so even shorter. This is, we think, a very exciting study. It's in our immune therapy refractory component of LungMAP. And this is looking at a promising new agent, which is a super agonist of IL-15, and that's this N803, together with pembrolizumab versus standard of care in patients who have previously had immunotherapy. So it fits within that framework. It comes out of work of Dr. Rengel's lab, and Hatem Hussein from UC San Diego is the co-chair. Hatem, do you happen to be on? No, he was on earlier, so, all right. Well, this shows the schema of the protocols. So again, prior checkpoint immunotherapy, now with disease progression. There are 2 cohorts, and I think this is incredibly important. And LungMAP has taken the lead out of all the studies that are being done in saying there's a big difference in people who have de novo resistance. They never responded, versus a group of patients who benefit, respond, and then subsequently relapse. So we're looking at these as separate cohorts. On the left, the primary resistance cohort. On the right, the acquired resistance cohort. And you can see both of these are randomized to the standard of care therapy versus the experimental combination. One thing I don't recall off the top of my head, probably some of you remember, is this our standard of care? Does it have a menu of drugs, or is it only docetaxel ramucirumab?
It's docetaxel ramucirumab,
I can't see
docetaxel
it here.
gemcitabine.
With the bottom
Or
of the slides are
pemetrexed.
cut off on this laptop.
Yeah,
All right, so actually you can see it.
Yes.
Okay, so you can see standard of care options are there.
so and just noting that the reason why the primary resistance cohort is a randomized phase 2, because there's less evidence to suggest that maybe they can overcome resistance. So we wanted to be more conservative. Well, the acquired resistance cohort is phase 2/3.
Got it. Important. Thank you, Mary. So primary objective is overall survival. And you can see the secondary objectives there. This investigational agent N803 is given subcutaneously every 3 weeks. Pembrolizumab is given in a standard every 3-week schedule. We already know if we have 900 altogether, already we have 170 sites with CIRB approval. So that's quite good, but we expect that to jump up. I know at UC Davis, we're hoping we activate it this week. Number of patients.
It's actually 5. We have 4 on the acquired resistance cohort and 1 on the primary resistance cohort as of yesterday.
Great. So even you can see day by day. We have to update the
Things are
slides.
ticking up.
So that's great. Great to hear. And just the acknowledgements from John. including, of course, our participating patients. So thanks to you all. And that brings us to Janelle, who I know is in the room. Janelle, I'll give you.
All right, so we'll have the what I call the refresh of the S1900B study. I think many of you are familiar. We had that study open, and then while it was open, selpercatinib got approved. And so we wanted to kind of take a step back and see what we could do to really answer some new questions that have been posed in that setting of that approval. And it really came down to, what do you do after you progress on a RET TKI that's now, and we now know there's more drugs approved in that space. And so we met with the lung committee and had a discussion as well as with the company. And this is kind of where we have landed in those patients who have already received a prior RET inhibitor therapy, that we will randomize them to receive chemotherapy with carboplatin and pemetrexed plus or minus selpercatinib. The primary endpoint for this is investigator assessed progression-free survival. We will have serial blood collections with liquid biopsies, including ctDNA, both on treatment, at progression, and at screening. And we think that by capturing the liquid biopsies at screening, we're actually capturing those resistant mechanisms to the single agent RET inhibitors, and then subsequently see what happens as the tumor evolution and so forth when you overlay that with chemotherapy or with chemotherapy alone. Our sample size is going to be 66 patients. We are CTEP approved for this new protocol. We just underwent review at CIRB and received no questions from them yesterday morning, so didn't have to get on a call, which is a great sign usually. And so we're hoping we'll get the comments, respond to those, and then hopefully be activated hopefully by June. And I'll take questions at the end. Thank you.
And next, we're going to go to Dr. Haas Borjai, who is virtual with us, who will talk about an update on the drug selection committee. As I said earlier, it's quite exciting. We have so many new proposals. So Haas, you're with us?
Yes, I'm here.
And if you can just let us know, we'll advance the slides for you.
Okay, great. Thanks. Thanks for the invitation. Glad to be here. So let's go to the next slide. Oh, these are disclosures. Next. So drug selection committee obviously is responsible for bringing in new ideas and concepts for consideration to be included as sub-studies for the entire LungMAP program. So as you have heard, traditionally we've had basically targeted or molecularly driven approaches for the management of of our patients with non-small cell lung cancer, and we also have immunotherapy approaches. And you just heard about a very exciting study that has just recently opened up, as Dr. Gandera was presenting. So we are looking at every possible way of treating lung cancer after initial treatment that patients are receiving. Obviously, again, both histologies are now included as part of the protocol. So what we do in drug selection committee is that investigators around the country who might have ideas about specific concepts, they can come to us, or our industry partners, if they have drugs or combinations that they think is ready for basically a phase 2 approach, they can contact us. And we bring these candidates in. We have several layers of discussion and basically help the investigators to come up with the concept that we think is suitable for inclusion as part of the LungMAP program. So key investigators, clinical researchers, investigators with interest in biomarker, the NCI, the FDA, everybody is involved. We have a scoring system after the review of the proposal that basically goes from scale of 1 to 9 on several different categories, as you see on the slide. And based on the comments and the scores, we either decide to proceed with the proposal or reject it or go back to the drawing board. And the process has been, I have to say, working fairly well. We get really good comments from everybody who's involved. Our colleagues at NCI have been very helpful. We had a very, very interesting discussion with the FDA recently. We're hoping that we can engage them even more and earlier in the process as we are thinking about specific concepts. So I think that the process has come together quite nicely to ensure that we have successful proposals and then hopefully positive studies that would help our patients. Next. As I said earlier, the substudy suggestions can come from a variety of different sources. Majority have come from our colleagues around the country, although we've had a few pharmaceutical companies approaching us. The drug selection committee, we have a core of more senior investigators from around the country who initially meets with either the investigators who propose the study or the industry partners. And then if we decide in this core group that the study is feasible or basically has legs to go on, then we proceed to a full drug selection committee review, Q&A, and then revising the proposal based on the comments, and then eventually going ahead with actually having a protocol initiated. Next. Right, you've seen this already, so I'm not going to necessarily go into this. You are all aware of the biomarker-driven sub-studies and the non-match. We've talked about that already. Next. So some of the key concepts that we've been working on. So we decided a little while ago to actually concentrate on the VEGF pathway for a variety of different reasons. We have 2 approved drugs currently, antibodies in this setting, VEGF inhibition, shown to be active in various types of non-small cell lung cancer. VEGF inhibition seems to be involved in a number of different pathogenic processes that are relevant to lung cancer. So there were a lot of reasons for us to want to go this way. And also, again, clinical experience with VEGF pathway has been rather positive. So we concentrated on this particular concept. So one of the studies that you see here, going after EGFR exon 20 insertions, a very rare patient population. Usually it's difficult to do single institution or even 2 or 3 institution type of studies just because of the rarity of the disease. So we are exploring EGFR exon 20 TKIs with or without ramucirumab in patients who've previously been treated with such therapies. And again, the rationale is there's an unmet need. We have a rare patient population and therefore a group like LungMAP with a number of different sites that are participating makes it an ideal scenario for us to conduct these sorts of studies. And again, the single agent activity of some of the agents out there is pretty reasonable, but it's not sufficient. So definitely improvements are needed. Put these together, the clinical needs together with the availability of drugs and the potential for biomarker studies, it just makes it an ideal scenario for us. Next is to go after MET, again, another rare patient population. Again, yes, there are some MET inhibitors, but the durability of response and all that is not quite there. And there's some preclinical rationale to combine a VEGF inhibitor with the MET TKI. And again, we are an ideal group and setting for going after this rare patient population. The other study that's on the bottom of the slide, and again, the formatting is a little bit different, so I'm not able to see the entire slide. This is for patients with MET exon 14 skipping mutation, is slightly different than the MET amplified group that you see in the other study. Next. For the non-match arm, again, this is a rather large patient population now in our clinics, patients who are receiving chemotherapy plus a platinum doublet, immunotherapy plus platinum doublet chemotherapy. Immunotherapy has been wonderful, has been great, but it obviously doesn't work for everyone. So again, realizing that there's an unmet need there and that patients usually have a pretty good performance status coming out of immunotherapy, with or without chemo, and therefore there's a need for additional improvement. We put together a couple of different studies so far. One you heard that's going to be presented hopefully soon, but this other trial, as you see here, a phase 2/3, a randomized study of docetaxel ramucirumab with or without cemiplimab, one of the newer anti-PD-1 antibodies, has now in the process of going through the protocol activation and all of that, and hopefully it will be a sub-study for us fairly soon. Again, for this patient population, after initial treatment, the options are limited. We also have the opportunity, obviously, to study the activity of some of the newer agents. In this case, TIGIT has received a lot of attention, showing some promising activities early on. So we have access to an antibody combination with pembrolizumab and a TIGIT antibody that might be an interesting target in patients with lung cancer. So study combining this dual antibody, so again, a TIGIT and pembro together with lenvatinib, which is an oral TKI for patients with advanced disease following frontline treatment, is under development. Again, I think this is also a very interesting approach, and if successful, it brings a very novel therapeutic combination into the clinical setting for our patients with metastatic lung cancer. Next. So there is a huge unmet need out there. I think we are well positioned as part of this group to be able to have a major impact in the care of our patients, having involvement from not only the pharmaceutical partners, but also other investigators around the country and NCI and the FDA. Makes this a very unique opportunity for us to be basically heavy hitters in the world of drug development, and again, to advance the science, take advantage of biosample collection that's going on as part of all of these studies, as you heard from Dr. Cousino, and therefore overall have a huge impact on the future of lung cancer treatment. And I think that's my last slide. Thank you.
Thank you, Haas. Now, one of the special things about LungMAP, which sets it aside, makes it unique, is that we have a number of other committees to help LungMAP be as successful as it can. And one of those is our Accrual Enhancement Committee. We'll have an update from Jennifer Pope. I'll give the advancer to you here.
All right, my name is Jennifer Pope. I'm here from the Yale Cancer Center, and I'm happy to give some updates on the work that the Accrual Enhancement Committee has been doing and our future directions. These are the members of our committee. As you can see, they are from a variety of LungMAP's partner organizations. The goal of our committee is really to work collaboratively with study leadership, the NCI, SWOG, operations and statistics, the study coordinator committee, lung cancer advocates, our collaborating cooperative groups, and also pharmaceutical company partners. Our committee, its main aim is to increase awareness of enthusiasm for and engagement in LungMAP among clinical investigators and site staff, advocates, and patients by fostering educational efforts around LungMAP to maximize accrual to the treatment sub-studies. One of the ways we do that is through patient engagement. So we are working on developing patient-friendly trial summaries for our new sub-studies. We're hoping that these will help patients make decisions about whether to learn more about LungMAP. We are also developing social media toolkits for new sub-studies, and these are going to include patient-directed messages and graphics, which you can see some examples of on the screen for S1800D. These are being drafted right now and in the process of being submitted to CIRB for approval and then use. We are also working on re-engaging our patient advocates. We have a patient advocate who sits on our committee. And at the end of March, we held our first patient advocacy webinar. And in that webinar, we discussed the important role that LungMAP plays, particularly in reaching community treatment centers. And LungMAP leaders presented the historical context of LungMAP and also the critical role that advocates play in the future directions that LungMAP will take. We are going to continue these advocacy webinars with patient advocates on a quarterly basis. And we are hoping they will be a key venue for fielding input from advocates on LungMAP priorities. And you can see this is a picture of our session. We also want to increase engagement with investigators and site staff. So we're doing a variety of things to improve this communication with both investigators and site staff in general. We publish a quarterly newsletter that gets sent directly to site staff and also gets posted to CTSU. We send thank you emails to site principal investigators when they reach accrual milestones. And we also send automatic email blasts out to PIs and head CRAs when a new sub-study activates. We have provider-directed tweets and a social media toolkit that will be provided for each new sub-study as they activate. And our committee really works in close partnership with the site coordinator committee to develop this content. They help contribute specifically to the newsletters and all of the work that we do. And we are really working to engage more regularly with that committee so we can make sure that our efforts maintain the site perspective in the work that we're doing. You saw this slide earlier today, but the Accrual Enhancement Committee is doing some work to support LungMAP's patient diversity and help contribute to its continued success. So right now we're working on reaching out to sites and also the study coordinator committee to get some information on from sites who have been especially successful in accruing a diverse patient population. So we can share that information with other sites. And we are also looking forward to distributing information on published results for LungMAP's patient diversity success when that is released. And I think last, I'll just end with our contact email. If people have questions, suggestions, or feedback, this is the email where you can reach our committee at. Thank you.
Thank you, Jennifer. And the last presentation before we go to the Q&A session will be by our Vice Chair, Jyoti Patel. Jyoti?
Great, thanks so much, David. If you could advance my slides. So really, this comes at the heel of what you can see is a huge amount of work that's been going on in these partnerships that have really solidified, I think, an infrastructure for us to be more nimble and to bring trials to patients. If you want to go to the next slide. So I think the future is robust. We have 9 years of effort of planning and executing, and now we're at, I think, a pivot point in which the science has really come to some maturity and enabled us to better select what we're doing. Go ahead and push return. I think one of our biggest opportunities that we have, certainly the advent of immunotherapies, has revolutionized what we do in clinic and how we approach our patients. But with this has really come an opportunity to understand and overcome resistance to immunotherapy. And so particularly for the non-matched arms, I think this is going to be more and more important. The correlative information that we have, the tissue as well as the blood, I think will provide many insights into looking at acquired resistance as well as primary resistance. And our group has been working on clearing those definitions in an effort to embed this in our non-matched arms. Return. We also have lots of opportunities to refine targeted therapy. So you've heard about a study in EGFR patients in whom a small group of patients developed MET amplification, and looking at VEGF inhibition in combination with those TKIs. Here again, we're able to really think about how we can use combination regimens. And then from the sotorasib trial, we know that further defining which populations would benefit the most from particular targeted therapies will be invaluable. Next slide. A return. We urge you to really come to us and to reach out to Dr. Cousino to think about how we can utilize the existing blood and tissue that we have, again, to understand best therapy. So this is a really unique repository, and now we're doing blood collection when patients come on study, and it's embedded in all studies when patients progress on therapy. And so again, here is an opportunity, I think, to really contribute meaningfully to the science and understanding of resistance. And one more. One of the biggest things I think Dr. Gray talked a little bit about was this is an opportunity for us to really collaborate among the different cooperative groups and to really develop clinical trialists. You know, we don't want to be sort of the unicorns anymore. The world has changed, and a lot of ideas happen outside of cooperative groups. But again, we think that it's absolutely valuable that we serve the patients that we see across the country from diverse backgrounds and in diverse locations. Again, LungMAP gives opportunities to more junior trialists to work with people who have sort of a machinery in many ways that's moving, that's well-oiled, but also to work with study champions across different cooperative groups to really bring these ideas to patients and then to really see the outcome of these interventions. One more. You know, it's absolutely important though, and I'm so glad we ended with Dr. Pope. Really, patients remain at the center of our mission. As Dr. Gandera has said time and again, one of the most amazing things we do in LungMAP is we bring the trial to the patient. So it's the right drug now with what we know about foundational science at the right time. So in the second line setting, and again, in the right place, which is where patients live. The work we've done more accurately mirrors lung cancer patients that you and I see in clinic every day, and really speaks to the diversity of our country, as well as access to therapies, therapy that may not happen only in academic centers. And so again, if we think about how we're executing these trials, how we're incorporating different biomarkers, how now we're using commercial foundation medicine to help guide where patients go. You know, I think we've done a lot, and that's essentially been work with all of you on the committee and our great partners in FNIH, FMI, the FDA. It's really been such a pleasure. So with that, I would love to take some questions, and anyone who wants to turn cameras on, please do.
And if we have any questions from our virtual participants, if you can put them into the chat, we should be able to see them there. If you're in the live audience, we would ask you to go to a microphone and state who you are. So Ray, who the heck are you? Where are you from? And what do you want to know?
Sometimes, you know. Ray Osaro-Gabmo. I'm the PI of a minority underserved NCORE, and I want to say, first of all, thank you. This is such a brilliant idea. This idea of bringing clinical trials to the patients where they are, where they choose to seek care, is where we really need to go. So LungMAP we are proud to say in the NCORE has been super supported by NCOREs, especially non-academic NCOREs, including especially the minority underserved NCOREs. So kudos to everybody. To have a question, 1800D. So there's an industry-sponsored trial called the QUILT trial that's using that same agent, N803. Patients randomized to the control arm of that trial, they ought to be eligible for the placebo control. It's unblinded, so you can tell at the time of progression who was not on the drug. Presumably they'll be eligible for 1800D.
Yeah, there's no reason to exclude those patients, provided they meet the eligibility criteria for our study.
Okay, that would be good to know, because it's a pretty large trial,
Yeah,
and I know it's become one of the go-to trials that we have been using. I suspect it's the same for a lot of other institutions.
And we were anticipating that we might actually even get to the answer for our study before QUILT becomes fully mature. So.
And Dr. Gray, your salpacatinib trial, is salpacatinib one of the RET drugs that can be used in first line because there's chemotherapy added to it?
Correct.
Okay.
Correct. Right. Exactly.
Other questions?
There's a question in the chat, I think that goes to Dr. Kozano. So Dr. Sang asks from Fred Hutch, for translational medicine, what recommendations do you have for junior investigators who are newer to SWOG or the other cooperative groups to understand sort of details around what specimens are available, if there's T cell receptor sequencing data available, really how to work with you effectively, David?
Absolutely. So just want to say welcome. And if you're a new investigator, I'd be particularly excited, even just to have a one-on-one call. Just email me. I'll put my email here in the chat, and happy to give you a bit of an intro to what we have. The T cell receptor sequencing data that we do not have, but of course there is great interest in such data. And so, the overall philosophy is that in the hopes that that data does eventually become available, that's the kind of thing that we would want to make available. We just don't have it now.
And Dr. Sang, you can talk to me since we're at the same institution as well.
Sounds great. Thank you so much for your help and questions.
Other questions? So what are we doing right? If you have a compliment, please come to the microphone and you can give it. If you want to say, what are we doing wrong, you can also do that, because as we've said several times, LungMAP has had to turn over basically multiple times since its initiation. You can imagine we went to all this trouble to initiate LungMAP, and about less than 1 year after we initiated, nivolumab was approved in the second-line treatment of non-small cell lung cancer, both for squamous and non-squamous. So we had to pivot and evolve, and we've done this several times, but we're always open to suggestions.
Well, Nagla Karim, Augusta University Georgia Cancer Center. So basically my question is, so LungMAP is strictly second line, where, for example, patients who are, like having a targetable mutation or, something like that, they have to go through the platinum doublet first and then something like that. Anyways, is there a chance that there could be some changes that it's not strictly second line? Like we see sometimes patients having a resistant, sort of like a mutation that just comes on top of, with the serial liquid biopsies and so on, or it's going to continue to be strictly second line?
So Jody, do you want to handle that political question?
Sure. So you point out very nicely that particularly for targeted therapies, the studies will be different. As you know, with the selpercatinib study, right? That's looking at platinum with or without the drug. For the frontline setting for non-matched arms, we have certainly been talking about this for some time. Our feeling has been that sort of screening patients, allocating patients to therapy, really getting the data from FMI in a usable format for us right now, I think precludes frontline therapy for most people, the timeline to move. There are a lot of, there's a large cooperative group study going on right now looking at Dr. Borgai's study, Insignia. We also have the SBRT study with any immunotherapy. So we also have to be cognizant that we're all competing against each other in some way. And I think at this juncture, our interest is really is understanding roots of resistance for immunotherapy in the non-matched arm and predictors of response. I think this is where we can really contribute something different rather than just pick a winner in the frontline setting.
But there's also, for example, Sarah Goldberg's study looking at MET amplification as a resistance mechanism for EGFR targeted therapy, osimertinib. So we're expanding to not just restrict ourselves to targeted patients who have exhausted all lines of targeted therapy to participate on LungMAP. So as David was saying, we're always evolving.
Other questions? Well, we're just about out of time. Any further comments? So Mary, you've been relatively quiet during this session. What other wisdom
Just like
can you give
usual.
us? I'm sorry?
Just like I always am.
Right. What other things should we bring up today?
Gosh, that's a good question. I mean, I think that
Talk about statistical flexibility.
Ooh. Well, what I was going to say is that, I mean, the thing that I think that LungMAP has exemplified, as you were pointing out, is that we have an infrastructure in place. And so having an infrastructure in place to be able to address finding better treatments for patients with lung cancer is an amazing thing. And we started working on this, well, we started talking about it in like 2012, and we started really building it in 2013, and we launched it in 2014. So it's been a major part of our lives for a long time. And so we have an amazing group of people, and that spans medical oncology, thoracic, just all kinds of translational medicine. I mean, the patient advocacy, everything. So my approach when I design studies is to basically, I'm the lead statistician, if you didn't know that, is to design studies that answer the questions and make certain that we're moving the science forward. And so that we always keep our eye on that is the point. And so when you keep your eye on the goal with respect to that, then you always just move the science forward. And we try to do that without being too hindered. And as you pointed out as well, I mean, S1900A, which will be presented soon, that study accrued substantially faster than we projected. And we expect that 1800D is also going to have that happen. So the more studies we have in the pipeline, the better off we are. And I think that LungMAP is just going to soar.
So I think Mary makes a good point in that there's always been a complaint from industry that CTEP sponsored trials take a lot longer to activate. And we're really trying to turn that around in terms of following very closely all the procedures and trying to do everything to get the study open. But once it's open, nobody has a network like we do. Nobody has 900 sites that are all, by definition, CIRB approved, or this mechanism where one study can open and close and another one's still ongoing. So we have some advantages, and I think that's starting to show, Mary. Let's wrap up. Janelle or Jody or Roy, any final comments?
If I can add, I think, fantastic meeting. And I want to touch on something that Dr. Kareem brought up. And I don't know if Dr. Goldberg, I'm going to pick on her too, because she's here today. She wants to say something about this. But, we've really started these discussions with Dr. Patel, Dr. Herbst, Dr. Vandera, with Mary, and with Ms. Pope here, that we need to start thinking about how are we going to think a little bit differently about LungMAP than what we have before, and how do we message that to the patients and the providers, right? If we're going to start bringing in these targeted therapies and start looking at them from the resistance standpoint, I don't think it's something that is a go-to for us to think about. And so just want to bring that to your attention as you, when you leave this meeting and go back to your sites to discuss these with your team and your team members. And the other thing is happy 10 years. Congratulations, right? You said 20, right? It was 20. I didn't realize it was
Well, that
2012 when you
that was
initially
the
presented
initial-
it.
Design.
Oh, yeah,
We
yeah.
had an initial meeting, but the launch was 2014. So
yeah.
we're not quite a teenager.
The thought and all the work that went up to getting this open in 2014 and all the excellence that has occurred. So is very impressive. Congratulations to you all.
Chanel, I really appreciate your charge to us. We need to be innovative, look for the newest things, look for resistance. So please bring us your ideas and concepts, and thank you. This has been a very nice hybrid meeting, David. Thanks for moderating.
Okay, well, have you already had your gumbo for the day, Roy, or are you going out tonight?
Remember, it's an hour, well, it's 2 hours later here. So we will be going out soon.
All right, so Roy is at the AACR, where we should congratulate him. He was on the board of directors of that organization. And Jody, you have the final word.
Well, it's 5:59 in Chicago, so you guys all have places to be. Thanks so much for your engagement. Continue to reach out. Take care.
Okay, thanks, everyone. You know, Chanel, I have to tell you that I've been so impressed by how you're handling this initial meeting and your approach, really seriously.
Oh, thank you. Well, thank you.
I mean, not that it'd be so bad, it's I just figured she'd be that. Yes, and she's doing a great job.